Right, so welcome to those who are new to the 18th Malaysian Research Insider session, and to those who have joined us last Saturday, welcome back. So I'm Apple, I'm doing a PhD in molecular plant science in University of Edinburgh. This science chat is co-organized by MBIOS, 100 Scientists of Malaysia, and our media partner, Science Media Center Malaysia. So let's welcome our scientist today, Luqman Fatah from Kyoto University. He's doing his PhD in genetics, investigating the role of a transposable element called line 1 in inducing immune response and its contribution to autoimmune disorders. So over to you, Luqman, if you want to share your screen.
Okay, thank you, Apple. All right. So I'll start my talk. Right. So then I went into I don't know why it's wrong with my internet connection.
Yeah, it's a bit choppy.
So I went to college at UM from A level. From 2009 and 2011. Then oh really?
Yeah, maybe just turn off the video to help.
Okay. Okay, so I hope it's okay now. So yeah, I did my BSc in Imperial College London. Then I went to King's College London for my master's, analytical science for industry. And then I become a research assistant at UKM. I actually have a 1-year gap in between, which I actually just mostly just chill and sell cakes. So I don't have any picture from UKM, so just put the logo of the company. And next is where I am right now. It's Kyoto University PhD in genetics, and that is my team. So in this talk, I will share about my research journey, more of how I arrived to what I'm doing right now. And I will start with the introduction of jumping gene or mobile element. So this element was firstly discovered by a scientist named Barbara McClintock. She was working on the DNA of the corn or maize. So she realized that some parts of the chromosome, the DNA material, appears in another different chromosome. And you can see in the left side in here. So the purple kernels, so normal C gene express the pigment product, you can see in the picture. But AC is the transposon, AC and DS. AC is the transposase that activates DS. And AC can transpose the DS into the C and disrupt this C gene, causing it to become yellow. And she published her work on 1950. However, during that time, the idea of DNA being able to be changed are not so accepted. So her work was not really being given attention only until 1960, where more people discovered this element that exists in all organisms. And later on 1983, she won an unshared Nobel Prize. So that's how she discovered the mobile element. Sorry, so now I want to start a poll. Sorry?
The poll is up. So how many percentage of the genome was considered junk?
Yeah, so let's see the answers.
Right,
Maybe
we'll give it
another
about 10 seconds. Yeah.
All right. So the correct answer is 98, which 35%. It's not bad at all. All right, so I'll show you the percentage of the junk element in the DNA. So previously, people only look at exons or protein coding genes, but actually in human genome, only 2% of the genes are protein coding genes. Only 2% of the genome. The rest, 98%, are considered as junk. And currently, we are still unsure what are the functions of these genes, and we are still trying to understand. And among these, 50% of them are the mobile elements. So line 1, ALU, SVA, others, LTR retrotransposon, DNA transposons are the retro elements, the elements that can jump. However, even though line 1 consists of 17% of the human genome and 500,000 copies, only around 80 to 100 could actually jump. And now I will show you the difference between different types of mobile element. There are two main types of mobile element, transposon and retrotransposon. So retrotransposon can jump through copy and paste mechanism, while transposon can jump through cut and paste mechanism. And the transposon, the DNA sequence is removed from the DNA, and the former insertion site would lose the DNA. And the same DNA would be inserted into a new DNA target. However, for retrotransposon, the DNA sequence would first be transcribed into RNA intermediate, reverse transcribed into a new DNA, and then this will be inserted into a new DNA target. And these are two transposed mobile element at new sites. So as the name suggests, copy and paste means that the original retrotransposon sequence would still be there in the same in the old DNA sequence. However, this would be removed in the case of cut and paste transposon. And some of the examples of retrotransposon are line 1, which I will be focusing on, and then retroviruses like HIV-1. And DNA transposons, for example, are ACDS, the one that I showed you before in the previous slide in the corn genome. Next, I would show you how line 1 jump specifically. So line 1 have two proteins, RF1 and RF2. So first, the full length line 1 will be transcribed to produce line 1 RNA. And then both RF1 and RF2 would bind to the line 1 RNA to form line 1 ribonucleoprotein complex or line 1 RNP. And this RNP accumulates in the cell, and they look like a small foci dot, as you can see in here. And then this line 1 RNP gets transported back into the nucleus. It creates a single strand break and then gets inserted into the genome. And as you can imagine, because the insertion, de novo insertion of line 1 could cause a lot of genetic diseases. And some of the earliest reported gene disruption reported are APC, which is a tumor suppressor gene. And this disruption of the gene by line 1 causes colon cancer. The other one is a factor 8, which is a gene important for hemophilia, which is important for blood clotting. So at first, when I first arrived, I don't really have research objectives. So we are pretty much have a lot of choices of what we are going to do. So my first idea is just to search for what kind of proteins bind to line 1 RNP. And there are a lot of host factors that binds to this line 1 RNP. So how do I know them? Is I just pull down the line 1 RNP by pulling down the RF1 protein using immunoprecipitation and then put it in the mass spectrometry. So mass spectrometry can identify what kind of proteins that are bound to the line 1 RNP. And then I got some hits. And if you have done mass spectrometry before, you would know that you would get tons of the protein hits. So you wouldn't really know what kind of proteins you want to be looking for. So while I was sifting through or looking through the results, I realized that there are a lot of antiviral proteins and some of the most common autoantibody targets in autoimmunity patients. So in the case of autoimmunity, you would have antibodies that bind to self-antigens. So when antibodies bind to this self-antigens, you would the cells, this would cause inflammation and kill the host cells itself. For example, in a situation where a viral gets into the body and into the cell, the B cell would produce antigen that binds to the virus. And in the case of a second or third infection, then the immune system would have adapted to the virus. But however, this system, for some reason, it targets its own body and kills the own host cells. And some of the autoantibodies that I found in the mass spectrometry results, the targets for the autoantibody are Rho, anti-Rho SSA, and anti-La SSB. So I'm wondering if line 1 has anything to do with autoimmunity. Then I check the literatures and I check the previous reported line 1 inhibitors. So what kind of protein actually inhibits or stop line 1 from jumping? And when I check the previous literature, I realized that a lot of the proteins that inhibit line 1 are actually antiviral proteins, and some of them are listed in here, like MOV10, ZEP, ADR1. And this makes me more interested in this topic. So I searched for more evidence, and some people suggested that line 1 might be the potential trigger for systemic or for autoimmune disease in general. So this paper suggests that, it shows that line 1 accumulation in the brain, in the neuron, cause neuroinflammation. And also another paper showed that line 1 induced immune response, which leads to the systemic autoimmune disease like systemic lupus erythematosus or Sjogren's syndrome. So let me show you one example. So in the case of one of the autoimmunity is called Aicardi-Goutieres syndrome. So in this case, it has a similar phenotype to a Zika virus patient, which is microcephaly, abnormal head circumference, as you can see in this slide. And interestingly, AGS or Aicardi-Goutieres syndrome mimics congenital viral infection, but when they check, there is actually no actual infection. But it has the similar effect as Zika virus infection, like high type 1 interferon, and the phenotype is very clear. And then I also check what kind of genes that found to be mutated in AGS patients. And I realized that there are 7 reported mutated genes, and all of them are antiviral factors that were reported to inhibit or regulate line 1. So the ones, these are the 7s that I listed. So they are IF1, TRX1, SAMHD1, and RNSH2. Next, I will show you what is the mechanism behind this. So this is innate immunity in the cells. So usually when an external or invaders like virus or pathogen gets into the cell, we have a sentinel or RNA or DNA sensors that would recognize this. So once the virus gets into the cell, they would release some materials that would be recognized by the sensors. So this would cause a stimulus in the cells, and this would release cytokines such as interferons. These cytokines are kind of a danger signals for the cells. And this is released to the extracellular parts of the cell, means it would get into other cells as well. And these interferons would bind to the receptor, and this would cause a downstream effect of activating the antiviral proteins or interferon-stimulated genes. So the expression of interferon-stimulated genes would inhibit this virus and remove these materials that are causing the interferon to be released in the first place. So in the case of virus infection, not so serious virus infection, this will only cause a transient inflammation due to the interferons. But what if the stimulus comes from inside the cell instead of external? So in here, we have line 1 instead as the endogenous stimulus. And once the line 1 gets into the cytoplasm, it would be detected by RNA or DNA sensors. And again, this would cause the release of interferon. And this would cause an expression of the interferon-stimulated genes inhibiting line 1 and removing line 1 from the cytoplasm. However, since this is endogenous and it keep being expressed, so more interferons would be released, and then the inflammation wouldn't, the source of the stimulus wouldn't be removed, and the inflammation would be persistent, and this will lead to autoimmunity. Next, this would arrive to what I am doing right now. So I'm wondering, what are the interferon-stimulated genes that are involved in this process? So if you remember from the previous slide, I still have the mass spectrometry results. So I just put the results from the mass spectrometry into the interferon database. So this database has the proteins that are upregulated by interferon. So I put this the results from the mass spec into this database to identify what kind of antiviral proteins are bound to the line 1. And I discover some of novel line 1 regulators, or ISGs, or interferon-stimulated genes. So to put things into perspective, I found some novel regulators, and I'm currently trying to understand the mechanisms of how these regulators inhibit line 1 and how they are also being expressed. So from this understanding, I hope to get more information or get a more comprehensive understanding of how the whole process actually happens inside the body. And last year, recently, 2 papers were published that kind of established the idea that line 1 drives interferon production. So one in Nature, the other one in Cell Metabolism. So in these papers, they used mice models, and they showed that increase of line 1 expression leads to higher inflammation. And this also contributes to aging phenotypes in the mouse. So in summary, line 1 consists of around 17%. If we want to include all of the mobile element, or transposon and retrotransposon, is almost half of our genome. However, not so much study have been done before, but now people are getting more interested towards the part, I would say, the junk DNA part that has been relatively understudied before. And line 1 has been shown to cause type 1 interferon response, leading to inflammation, autoimmunity, and aging phenotypes. So that's the end of my talk, and thank you for listening.
Wow, amazing. That was a very cool talk. Thank you.
Thank you.
I was just interested. So how is line 1 jumping triggered normally?
Normally?
Yeah.
So in our body, we still have active line 1. There are around 80 to 100 copies of line 1 that are still active. And in somatic cells, they are actually usually inactive because of hypermethylation in the CpG islands. However, in some other cells, interestingly, in some other cells like brain cells, there are still some ongoing active line 1 jumping. And this has actually been proposed to be one of the reason of mosaicism of the brain. So it keeps getting a different genetic, I would say, genome profile due to the jumping of line 1. But this is in human, but in other mammals like mouse, they have a lot more of the active line 1.
Yeah. So does stress induce a higher expression of line 1?
Yeah. So a lot of stresses have been shown to induce line 1. One of them is UV that we have talked before. I'm not really sure why the stress itself. Why the stress itself actually causes an increase of line 1. But some hypotheses, some people think that I also think along the same line is that having a mobile element in the genome is kind of a high risk, high reward thing, where in terms of in the stressful situation, you might want to have more options of what you have in the genome. So probably it can cause a lot of diseases if it doesn't occur, it doesn't jump properly. For example, if it jumps into a functional gene. But if it can create, for example, a new gene or rearrange the genome in a beneficial way to the host, so that would be beneficial in terms of survival. So that's the kind of evolutionary view of why stress increase line 1 expression. But in terms of mechanistic view, there are, I'm not really sure, but there are some involving the mostly methylation of line 1.
Yeah. So something on top of my head, I was just thinking, let's say if you said in somatic cells it gets repressed by factors, right? But
yes,
obviously there will be cases where the line 1 becomes active and jumps into an important gene. Does that ever happen? And if
yes.
Yeah, and if it does the cell just kills itself by apoptosis?
Yeah. So it's kind of how, I guess, it's also one of drive for carcinogenesis. So in a lot of cases, most cases, the cells would apoptosis, but if it survives and if the cells get some kind of, I'm not sure, some kind of mutation that can cause it to become more immortalized, so that might cause it to become cancerous. Yeah. So
is there any research showing that line 1 can cause cancer?
Oh yeah. So I showed in the one of the maybe I was too fast. So it the first report was on 1992. It jumps into APC gene. So APC gene is a tumor suppressor gene for colon cancer. Usually colon cancer patients have disruption in the APC gene. And yeah, so currently there are a lot more reports on line 1 being inserted in tumor suppressor genes, but not just cancer, a lot of other genetic diseases as well, like hemophilia.
Yeah, that's quite cool, because I think the general public always thinks that it's always external factors that makes you get cancer, but sometimes just internal factors like a jumping gene could
you couldn't really, I mean, even if you like take care so much of your diet, it's a lot more to do with genetics. So
it's
really
or just chance,
Yeah, it's chance. So it's very difficult.
So apart from autoimmune diseases, are there any other diseases that are associated not just with line 1 but other transposable elements as well?
You mean other kind of diseases?
Yeah. Apart from the one you mentioned, which is systemic lupus.
You mean
erythematosus.
autoimmune diseases? Yeah.
Mm.
So I showed systemic lupus and Sjogren's syndrome autoantibodies, and I think the more obvious is AGS. Most of these autoimmune diseases names are very difficult
Yeah,
to
yeah, I know.
pronounce. So
Yeah.
Aicardi-Gautier syndrome (AGS).
Mm.
So they are reported on which genes have been mutated, and in total, people have found 7 genes, and all of these genes are actually line 1 inhibitor. So it's pretty, I would say, in hindsight, it might kind of obvious. And also, one paper actually showed that in mice, when they upregulate line 1, it has the similar AGS kind effect where a lot of the cells apoptosized, and this caused the mice to have a smaller head called microcephaly. So the
Oh, wow.
same as a Zika virus and AGS patient.
Right.
Yeah.
Oh, I didn't know you can see that in a mouse model, but that's quite cool.
Yeah.
Yeah. So on to the next question. A question by Neural. Do you think that SLE would be curable in the near future? And that kind of relates to my question, like, is there any possible therapies against line 1?
For now, the field is pretty new, I would say. So, but I checked some companies. One company is actually developing. It's not line 1, but endogenous retrovirus. It makes 8% of the genome, and it's a similar mechanism as line 1. So they are currently making the inhibitor of endogenous retroviruses. But in terms of a cure, that's a very difficult thing to do. I'm really thinking hard about it right now on what part of the mechanism can we actually tackle, so we can actually either reduce the phenotype, reduce the inflammation, or totally cure. Totally cure might be very difficult, but at least if we can reduce the inflammation. So for now, there is no cure for autoimmune diseases. Usually what the drugs that we have right now are usually the anti-inflammatory drugs.
Yeah.
So it's either they reduce the interferon level, or usually they just block some of the proteins that are involved in the interferon receptors. But in this case, it's kind of dangerous because you would lose the ability to adapt to viruses, or at least you would have a very weak immune response towards viruses. So I don't think in long term that's a really good plan. I mean, for now, yeah, at least we can reduce the effect on the patients, but that's not a cure at all.
Yeah, it's quite tough to find a cure to just stop line 1 from jumping, really. Cool.
It's because there are a lot of line 1 in the cells. So if it's not like one gene where you can just target the gene with a CRISPR and then just repair it.
Yeah.
Even then, that's still very difficult because you have billions of cells. But yeah, in terms of line 1, it's not impossible, but it's very difficult. I mean, you can use like a siRNA or antisense oligonucleotides to block it.
Yeah.
So yeah, I'm also thinking of that.
Yeah.
So how can we actually inhibit line 1?
So in addition to that, like, what are the sequence similarity between these line elements? Are they
similarity?
Yeah.
You mean like why some of them are active, some of them are inactive?
Well, I guess that's just like genetic sequence, like
yeah. So
are they exactly 100% the same, or?
Oh no, they are all they're all different. But the similarity, there are multiple I would say multiple clades or multiple types of line 1, and currently the one that is active is only one type, which is called line 1 HS. And there are a few types, but this one is the most active. and it's the host and viral interaction hypothesis, where once the virus gets into the cell, the host would try to inhibit or stop it from proliferating, from increasing their copy numbers, and then the host would survive, and then most of them would get inactivated. But then the virus would keep getting mutations, and then the host has to adapt. And so this is like a combat between host and virus.
Yeah.
So it's an ongoing kind of ongoing battle. So that's why we only have a very few active line 1 elements in the cell.
Yeah, cool.
Yeah, but
Yeah, so Jake mentioned that, you showed us some line 1 papers in aging models. So for your research, was the line 1 mass spec done in aging models or senescent cells?
No. So my research is it's not very, I don't use animal model, so it's just cells on plates. So for now, for my PhD, I'm focusing on just the regulators and the mechanism. But maybe later I'm also thinking of mouse models, but not in the near future. I'm not really, not in the near future.
Yeah, that's cool. So Jake also asked another question, like how can we target line 1 specifically for SLE with siRNA? Is it through the RNA intermediate?
Yes. So okay. So in terms of the line 1 itself being detected as the virus, few papers showed that it's actually the RNA being detected, and some other papers show the cDNA being detected, because line 1 also has the reverse transcriptase, so it can change the RNA to cDNA. So in our in the cells, in the cytoplasm, we have DNA detector and RNA detector. The DNA detector is not specific, so any kind of DNA in the cells would actually be detected, and then it would induce immune response. However, the RNA has some kind of criteria that they need before they actually bind. But both of these RNA and cDNA has been detected, but of course it needs to be transcribed to RNA first to get into cDNA. So I guess if we want to inhibit it, so it would be best just to inhibit the RNA. So we wouldn't have the cDNA at all.
Yeah, cool. So onto a more technical question by Sabrina. So she was wondering if you would be interested in looking at DNA methylation pattern change and line 1 jumping in SLE. Or is DNA methylation not really forte?
Because this is kind of funny, because the proposal that actually sent to get into this place is DNA methylation, but now I'm not doing DNA methylation at all.
I see.
So it's kind of funny, but DNA methylation is also a good way to see if it's activated or not.
Yes. And also for RNA methylation as well that she mentioned, because other one that you've shown on your slide was RNA editing enzyme. So
yes.
Yeah. Cool.
Yeah. So
just unfortunate, just a bit unfortunate that you weren't doing anything with methylation, but
yeah. That was my current previous interest, but I guess it's all right.
Is there a reason?
Not really, because it's not a scientific reason. It's more of
whether it's feasible, whether it's doable.
Yeah. It's more of a practical reason, because it's much easier to do what I'm doing right now rather than DNA methylation. It's just that the lab is not well equipped to study DNA methylation.
Yeah. Sabrina's quite sad. He's like, why, Luqman?
Maybe later.
Yeah, maybe for your postdoc or when you open your own lab.
There's so much that I want to study.
Yeah, exactly. So Jake asked, why Japan for a PhD?
I'm still thinking.
You still don't know after 4 years there.
So I've been in the UK for 5 years. Then I think that maybe I want to try something different. So I think I'll just gonna try in Asian countries instead. So I looked to Japan because it's one of the top universities, and especially Kyoto University, they have a lot of like Nobel Prize winners. So that might be a good idea to just try. Yeah. So then I applied to Japan. It's more of a gut instinct, I would say. I just set my mind on it and I go for it. Like there's, I wouldn't really say that there is like a real strong reasons for why Japan. Yeah.
Gut feeling. Sushi,
Food.
food,
Actually, we don't
anime.
really eat sushi that much in here. Anime,
Oh,
yeah, but
it's expensive, isn't it?
it's sushi is more like a weekly or a monthly thing. It's not the common food in here. Actually, I mean, it's not the food that you eat every day. Yeah.
So, since you've been there for 4 years, so how is it like studying in Japan, or just like the general research culture there?
Research culture. It's research culture is always depends on the lab, right? So it's kind of hard to say specifically, but I would say in general, they are maybe not so much collaboration. As the main reason is that I think because of the language, most Japanese can't speak English, and because of that, I also struggled very much, especially during my first year. So for example, when I go to the office and like alone, I have to just like sign language to
Oh no.
to try to talk. I was like,
yeah. So
although I
their
don't really
science, their scientific terms are there. Do they use Japanese?
Totally, yeah, they use Japanese. So for them, it's also very difficult. So for now, I can catch a bit, but for the new students, they're always scared as well to use English because it's they learn everything in Japanese, all the scientific terms.
Yeah. So Jake asked whether you picked up Japanese as a language.
I can speak the daily conversational, not anything advanced. Yeah.
Oh, nice. That's pretty good. As long as
Yeah,
your conversation, I think that's really good.
Yeah, like to random, not really, random strangers. It's kind of a no. I mean, you don't really talk to random strangers, but yeah, it's mostly to your colleagues. Yeah.
Yeah, so I don't think we have any more questions, but this is just a question that we end with every Research Insider session. So what's next after your PhD?
So, well, my ambition is I want to start a research company, but for now, I'm not really sure if I should just jump directly. Now I have a proposal, but I think it's kind of risky to just go blind. So maybe I'll take another position first, like in a company first or academia first.
Yeah.
And from there, I can get some networks and get some more ideas on my proposal, my research to start. Yeah.
Are you planning to open a company in Malaysia or Japan?
Well, I would love to open in Malaysia, but that's I'm still looking through it, so I'm not really sure which country would be best. But I think because there are a lot of science graduates, I think, in Malaysia. I don't remember the statistics, but actually we have, in terms of ratio of population to science graduates, we actually have quite a lot. But we don't really have a lot of job opportunities or like research that's research related. A lot of the job opportunities for science are, for example, for sales, distributors. Yeah, so it's very little of research in here. So I would like to like contribute to that. So instead of complaining about how lack we have, so maybe I can be one of the solution and help other Malaysians as well.
Yeah, and interesting. Yeah, like I'll say, yes, let's push the government for more funding instead of just like complaining all the time. Yeah. Right. Thank you, Luqman, for the amazing presentation and the great answers. And thank you for the audience for joining us on this Saturday. So our scientist next week is Joshua Koo. He's doing a PhD in cognitive neuroscience in University of Nottingham. and he is also one of the co-founders of 100 Scientists of Malaysia. So for more updates regarding future Research Insider series, like our Facebook page and bios and 100 Scientists of Malaysia. And see you next Saturday. Bye.